Yes, good day, class. Thank you so much for tuning on Metric Hub. I can see the participation, I can see the involvement, I can see the engagement. Thank you so much, and that's the spirit when we are working towards end of the year. Today's paper is 2022 November. It's a full paper that I'm gonna run through with you. Paper one. I'm not going to waste any time. Let's quickly just go through the paper and then we answer all the questions as quick as possible. I'm gonna just try to make it quite short. So the instructions, I'm not gonna go through them. You know always that when you go through, when you have to write exam, you always have instructions. Please note that you always read the instructions and follow instructions per question. Uh, let's go through the paper section A. It is multiple choice. One other tip that I always want to give to students and to you guys is that please make sure that in multiple choice and in any paper where you are given options, you don't know the answer, rather make any guess answer that you can give than to leave the answer blank because you will be actually like taking chances with marks. So just try to give any answer that you know. I mean, just find any guess answer that you can give there and like to leave blank spaces. It's actually not a good thing to do when you're writing exams. So question 1.1, which of the chemical equations below represent the process of photosynthesis? You see, actually in this question here, it actually wants you to understand the uh, process of photosynthesis. Basically, to know what are the requirements, what are those factors that are necessary for photosynthesis, and also the end products of photosynthesis. So, as you can see, we are saying the chemical equation below represent the process of photosynthesis. We should know that it is always carbon dioxide plus water plus energy plus light energy is equal to starch and oxygen. Starch and oxygen are always produced. So what I'm saying is that question 1.1, the correct answer is what is B, carbon dioxide plus water. We always know that during photosynthesis, carbon dioxide is used. It's actually one of the requirements. I remember the previous question paper that we did, which was an experimental question, where people argued about the answer. So please, the correct answer here is that it is B, the chemical substance used to extract chlorophyll from leaves in is dash. The chemical substance that is used to extract chlorophyll from leaves, it is what? It is alcohol, which is letter C. Our C is the right answer. Alcohol is used to extract chlorophyll. I'm sure again, if you remember, in terms of this touching a leaf, when we are testing if chlorophyll is necessary for photosynthesis. And then question next, which of the following raw materials required for photosynthesis is controlled by stomata? We are saying the opening and the closing of the stomatas controls what? Number 1.1.3, it controls carbon dioxide gaseous exchange. So raw material, the question is raw material. But otherwise, they can also say is the end product of photosynthesis. Therefore, it was supposed to be carbon dioxide. I mean, it's supposed to be oxygen. So carbon dioxide is a raw material. Hence, we are saying the correct answer is what? Is A, as the raw material. Please note me right. I'm saying if they say the end product it's a product of photosynthesis, not that it is oxygen. The main function of chlorophyll is to dash. The main function of chlorophyll is to absorb light. The main function of chlorophyll is to absorb what? It is to absorb light. The main function of chlorophyll is to absorb light, sunlight. I'm sure that you know, and we can actually confirm together that is why plants are green. And then which of the following stages of photosynthesis releases oxygen gas? Which of the following stages of photosynthesis releases oxygen gas? It is light-dependent phase. Light-dependent phase, it actually releases oxygen, which is letter C. Light-dependent phase is the stage of photosynthesis where oxygen is produced. Which of the following digestive juices contains no enzyme? Digestive juice contains no enzymes. I'm sure that you remember enzymes are catalysts. They actually speed up the rate of reaction. So in this case, which juice? Digestive juice does not contain enzymes. I'm sure you can agree together that it is what? It is bio. Bio does not contain enzymes. Number 6, it is C. The bio is the digestive juice, which does not contain enzymes. Number 7, study the list of processes associated with the human digestive system. They are associated with the human digestive system. So we have got detoxification, diminution, emulsification, and then peristalsis. The question is saying, which of the following combinations describe the digestive functions of human liver? Human liver, the process that it is involved in. Number 7, we are saying it is detoxification. It is correct. And then 2, diminution. It is correct. And then 3, emulsification. It is correct. And then the liver is not involved in what? In peristalsis. Peristalsis, it is actually taken, or it is a process that takes place through the gut and also the intestinal stomach part. They are the ones that do peristalsis. So peristalsis is not involved. Hence, our answer is what? It is C. It is C. The process is associated with the human liver digestive system. It is detoxification, diminution, and emulsification, which is C, number 7. Number 8, the epiglottis prevents the flow of dash. Number 8, it prevents the flow of food and water into the trachea, which is correct. Number 8, it prevents the flow of food and water into the trachea during inhalation of air into lungs. During inhalation of air into lungs, the volume of the thoracic cavity increases and the pressure decreases. The volume of the thoracic cavity, it increases. Inhalation, it increases and the pressure decreases, which is also correct. That the answer is what? It is A. The answer is what? Is A. And then number 10, which of the following tubes transport urine from the kidney to the urinary bladder? The tube that transports urine from the kidney to the urinary bladder, it is also the ureter, which is letter A. And then the ureter, it actually transports urine from the kidney. It transports urine from the kidney to the urinary bladder. It is A. So section A, I'm sure that we can agree that it is possible to get the 20 marks. Let's quickly just go to the next question. Give the correct biological term for each of the following descriptions. The correct biological term for each of the following descriptions. Write only the term next to the question number. So we just need to write the answer here. Don't write the statements. We write only the correct answer, the biological term that defines the process in question. The part of the chloroplast in which the light independent reactions of photosynthesis takes place, it is the stroma. I'm sure that it is repeating this question. We once answered it also in the previous paper that we did here. Next question, the cell organelle in which ATP molecules are produced during cellular respiration. It is the mitochondria. Mitochondria, we can say the mitochondrion or the mitochondria is the same thing. So it is actually the organelle in which energy molecules are produced during cellular respiration. It is where energy molecules are produced. And then the stage of cellular respiration during which two pyruvic acid molecules are synthesized. They are synthesized during what? The glycolysis. Glycolysis Glycolysis is the stage of cellular respiration during which two pyruvic acid molecules are synthesized. A single-celled fungus used in the making of bread, it is what? It is the yeast. Yeast is used for fermentation. It is used for making bread. Uh, the two main branches of the trachea we call them the bronch. It's the two branches of the trachea. And then the region of the kidney where renal pyramids are found, they are found in the region of the kidney where the renal pyramids are found. They are actually found in the medulla. Number 6, they are found in the medulla. Next question, the removal of metabolic waste products from living organisms is called excretion. When waste is released, removal of metabolic waste products from living, we call that excretion. It's excretion. Excretion is the removal of waste products from the living organism. The structural and functional unit of the human kidney, it is the nephron. Structural and functional unit of the human kidney is the nephron. That is correct. So I'm sure again we can go together and confirm that these are the correct biological terms for the statements that we are having in this paper. Let's quickly go to question 1.3. Where is it both? Is it A alone or it is what? So the mesophil tissue, it is the mesophil tissue. We are saying it is the palisade tissue. It is the sponge tissue, which means again, I'm sure that these are all correct. So it is what? Both A and B. The phase of cellular respiration during which carbon dioxide is released. Cellular respiration is released during the Krebs cycle, which is B alone. We are saying during cellular respiration, carbon dioxide is produced during what? The Krebs cycle. 3, the double membrane covering the lungs. The double membrane covering the lungs, it calls the pleural membrane, which is also B only. It is also B only. So let's enjoy the paper. We continue. I'm sure that you are actually seeing the relevancy and It is really making sense and sticking. Let's go to the next question. The diagram below represents the structure of the alveolars. And then we are seeing blood flow. We have got our blood flow. We have got our structures A and B. And also we have got moisture. We have got our structure C. And then we have got the in and outside of gaseous exchange there, which is showing uh, the gaseous exchange there. And then let's just go through, see the question. What does it say? Also, identify the gas represented by C. What gas is represented by C? I'm sure that you can see that at C, from the alveolar C, it represents what? C, it represents oxygen. It is oxygen that is being represented by what? By C. And then process at E, the process at C, at E, it is called diffusion. Diffusion, which we call that the movement of gas molecules through a concentration gradient from higher to low concentration. So E is representing what? Diffusion. E is representing E, our E there, it is representing what? It is representing, it actually represents diffusion. And then the next question is saying, give the letter and name of the part that transports deoxygenated blood, the letter and name of the part that transports deoxygenated blood. I'm sure that before even you go to the diagram, you should be able to know that where do we have oxygenated blood, where do we have the deoxygenated blood. So we are saying give the letter and name of the part that carries that transporting deoxygenated blood. The deoxygenated blood, it is carried through what? It is carried through the pulmon. You need to know that before I'm saying, before you go to the structure there, you are expected to know that it is the pulmonary artery or the pulmonary arteriole that carries what? That carries deoxygenated blood. So when you look on the diagram, you should see to say that it is letter what? It is letter A. It is A that transports deoxygenated blood. So letter transport deoxy. So we are saying A, which is also called the pulmonary arteriole. Or we can call that the arteriole. And then the letter and name. This question, please, class, note that most times you find that learners, you make a mistake of not answering the full question. So you need to give the letter and the name in this regard. Don't give only the letter and leave the name. So you are supposed to give the letter. The letter is A, and then it is the pulmonary or the arterial. And then the letter and the name that contains hemoglobin. Hemoglobin is actually contained in the what? In the red blood cells. So you need to look on the diagram to say where do we have the red blood cells, which we are saying it is also called the what? The erythrocyte. So we need to look on the diagram to say which letter represents the red blood cell. Where do we have the red blood cell? So on our diagram, we are saying the letter which contains the red blood cell. So we are saying it is what? It is letter D, which is we are saying which has got hemoglobin. So hemoglobin is contained in what? In D, which we are saying it is the red blood cell. We can call that the red blood cell, or we can also call it what? A erythrocyte. So we are saying that is the letter which contains the hemoglobin, D, red blood cell, or erythrocyte. That is the correct answer. And then number connected to the pulmonary vein, the letter and name of the party connected to number C, the letter and name of the party connected to the pulmonary vein. The pulmonary vein is actually connected to the what? To the venue, or we can say the pulmonary. So we are saying the letter and name that is connected to the pulmonary vein, it is actually the what? The venue, which we can have to look to say which letter is it. It is what? Letter B. It is letter B class. On question number C, we are saying it is letter B, which is actually the what? We can say it is the venue, or others they can say they also put it as to say it's pulmonary. You understand? So connected to the pulmonary vein, it is B, which you can see on there to say that it is connected to the pulmonary vein. So it is B, as you can see there. Name the tissue that lines the alveolus, the tissue that lines there, the tissue which lines number 3.1.4.3. It is called the what? It is called the squamous epithelium. I'm sure that we remember that if you can refer to your notes again, you can actually believe with me that the tissue that lies the alveolus is called the what? The squamous epithelium. You understand? And then name the tissue that lines the blood capillary. The tissue that lines the blood capillary. The blood capillary, obviously, on letter B, I'm sure that we agree that, or we know that it is called the what? The endothelium. That is the tissue that lies the what? The blood capillary. So, classy, take note of this before I move forward, or before I move to the next question. So, these are the scientific or the right answers that you should give to this question. As you can see, the structure of the alveolus. Okay, next question. The diagram represents a part of a human digestive system. Human digestive system, which is given there. We are given part A, B, C. We are given part A, B, C. So uh, the question I'm saying, I don't want to give the answers yet, but just look on the diagram there, and then it is saying identify part A, B, C already. Okay, we're saying identify the part A, B, C. Do you see again, class, that this question is repeating? So as you are revising and preparing for your exams, not as you are that examiners or your teachers and whatever our syllabus wants us to understand this. So A, it is the esophagus. That is A. And B is the stomach. And then C is actually the what? It is called the pleuric sphincter. Or others, the other books or other, we can just put it as the what? The pillars. So that is ABC, esophagus, the stomach, and I'm sure that you can actually not miss any marks there. Question next, that is ABC. I just answered them on the diagram there. Okay, state the function of letter C. State the function of letter C. The pleuric sphincter. What is it doing? Actually, it controls the movement of what? Of chyme from the stomach to the what? To the intestine. It controls the movement of the chyme from the what? From the stomach to the small intestine. That is the function of C. C controls the movement from the stomach to small intestines. That is the function of part C. Controls the movement of the chyme from the stomach to the small intestine. That is it. And then name the digestive glands found in the mucosal layer of B. The digestive glands found in the mucosal layer of part B. What are those digestive glands that are found there? Number 5, I'm sure that if you can refer very well, you know very well that B is the what is the stomach. So in the stomach, the digestive glands that are found in the mucosal layer of B, it is the what? The gastric juice. I mean the gastric glands, sorry. It is gastric glands are the digestive glands that are found in part B is the gastric glands, and then acid secreted by glands are mentioned in above. What is that? I'm sure that the gastric glands, it is responsible for helping what is it, chemical digestion. So what is the acid secreted by these glands, which is in B? It's hydrochloric acid. Hydrochloric acid. Hydrochloric acid is actually a digestive acid that is helping in chemical digestion, that is uh, secreted from the gastric glands. So section A, I'm sure that you can see, and then for sure 50 marks. I always in my from my class and everyone that I'm teaching right now, section A class, there is no way that you must not get less than 37, or I mean less than 47. If you get less, get 47 if it is not total. Because section A, you need to get all the marks right and don't make any mistakes. That's it. Next question, section B. The diagram below represents part of the human kidney, is part of the human kidney, and also we are just quickly going to answer the questions that are there. Part of the human kidney. So first question is saying, name the part shown in the diagram. Name the part shown in the diagram So what part of the human kidney is shown in the diagram here? What is it? It's called the what? The renal capsule. I can say that this is a renal capsule. We can say the renal, it is the renal capsule that we can say. And then the next question is saying, name the process that takes place in party represented in diagram. The process that takes place there is actually like that is taking place there. The process that is taking place, we can call that it is called what? The process that takes place in the party represented in the diagram. It is filtration. It actually shows filtration. Others, or can say ultra filtration. It is the process. Question B, ultrafiltration, and then identify part A, B, C. Identify part A, B, C. Part A, identify part A. Part A is actually called the afferent atrial. Part B, you can say it's called the what? The glomerulus. And then C, it is actually it is actually the glomerular filtration. The glomerular filtration. Those are the parts A, B, C that are asked there. The glomerular filtration, the afferent atrial, as you can see on what I've labeled on the page there on the body. Let me go to the next question. Or identify part A, C, and D. Part A, C, and D. So I already mentioned, I already labeled B and, sorry. The question wanted to identify part A, C, and D. Excuse, we are saying part A. Part A, I've already said that it is the afferent atrial. And then B, we said, C, sorry, is Then obviously D is the glomerular filtration. Yes, as you can see the arrow which is showing, we are saying the filtration is taking place at D, the glomerular, the glomerular filtration. Explain why high amount of protein in the urine indicates kidney damage. Why high amount of protein in the urine indicates kidney damage? 2.31. Uh, One thing that we need to understand is that proteins actually, protein molecules are large molecules. Protein molecules are large molecules. Protein molecules are large molecules. That's one thing that we should understand, which are generally not fast, which generally cannot pass through the what, which can generally cannot pass through the tiny pores of the thin glomerula, ne? Not passing through the tiny pores in the perineum. Into the capital space. So therefore, high amount of proteins in the urine indicates what, malfunctioning of filtration process. It indicates malfunctioning of the filtration process. Filtration process. So one thing that you should understand is that it is not possible for protein molecules to just pass through because of their size. So if they are found there, it means that the filtration process is not taking place properly. That is what we are saying. We cannot find protein molecules. Then when they are there, when we find the protein molecules because of their large by their large molecules in nature, therefore it means that the time it is not yet, I mean the filtration process is not happening properly. As well, also we can also talk about the tubular reabsorption filtration process. the, the tubular reabsorption. More so, I can also maybe mention that a tear in the grammar in the glomerular membrane also can lead to the what to proteins appearing in the urine as well. A tear in the glomerular, a tear in the glomerular membrane also it also can also lead to what to protein appearing in the urine and as well cause proteins to appear in the urine. To appear in the urine. That is the reason why question 2.1.3, why high amount of proteins in the urine indicates damage. It is therefore the reason because of what I'm indicating or I'm showing you here. Question 2.4, name the essential nutrient that will not be reabsorbed from the content in D in diabetes patients. We call that diabetes mellitus. So name the essential nutrient, essential nutrient that will not be reabsorbed, that will not be reabsorbed from the content of D from this glomerular filtration. This from the filtration there, glomerular filtration, we are saying what is this essential nutrient that will not be reabsorbed from the content of D? It is diabetes patients. We have got to understand that is called what? It is glucose. It is glucose that is the nutrient that will be that will appear, that will not be reabsorbed from the content of the glomerular filtration. And then question 5, name 3 ways in which the part shown in the diagram is structurally adapted to perform its function. How is this structure is structurally suited or adapted to perform its function? So what is happening here is that explain how the kidney is structurally suited to perform its function. Explain how the kidney is structurally suited to perform, which you should understand that specifically the renal capsule. So how is the renal capsule structurally suited to perform its function? That is what you should talk about here. Question 2.1.5. One, we are saying, how is this structurally suited to perform its function? So I'm just going to use a different pen for us to see clearly. We have got one, the afferent arteriole is wider than the efferent arteriole. That is one first point. Is wider than the efferent. Is wider than the efferent arteriole, or we can say that is wider than the efferent arteriole, or we can also say that the narrow the diameter of the efferent arteriole resists the flow of blood from the wider afferent arteriole. The narrow diameter of the efferent, the narrow diameter of the efferent arteriole actually resists, it resists the flow of blood, from the wider from the wider afferent arteriole, which actually causes high blood pressure, causing high blood pressure in the what? In the glomerulus arteriole, causing high blood pressure, which we said see in the diagram there, which causes high blood pressure in there. And also, we can as well, we can need also to understand that the glomerulus provides large surface area so that more plasma or liquid is able to filter through the endothelium, which within a short period of time. That is another point that we need to take note of. Number two, the glomerulus provides large surface area. It provides large surface area so that more plasma or liquid is able to filter through the endothelium. It is able, within a short period of time, is able to filter through the endothelium within a short period of time. So that is how this question must be answered. The question, don't forget that the question is saying uh, ways in which the part shown in the diagram is structurally adapted to perform its function. So what you need to know is that when you look to the red capsule, when you look onto this diagram, you must be able to see that what is it that is taking place, what is it that is what is it that is taking place there? So therefore, you see the structure, how is the structure assist to perform its function. We don't talk about anything else, but but only to explain the functions that are performed uh, by the what by the renal capsule to say how does it work as shown by its function. So ways in which this part in the diagram is structurally adapted to perform its function is what is answered on question 2.15. How is the structure in diagram the red caps the renal capsule, how is it structurally suited to perform its function? These are the points that you should give, and I'm sure that you will never go wrong. You will never get anything less than the best. So let's just go to the next question. An experiment was conducted to determine the effect of light intensity on the rate of photosynthesis. Procedure was as follows. Water plant of Elogia species were placed under a glass funnel in a beaker containing water. Test tube containing water with no air bubbles was fitted over the glass funnel as shown in the diagram below. 2 pinches of bicarbonate of soda were added to the water before the start of the experiment. After switching off all the lights, a light a lighted table lamp was placed 1 meter away from the beaker. A meter scale was placed between the table lamp and the beaker. The number of air bubbles released by the plant in a minute period was counted and recorded in a table. The above steps were repeated at different rates. Repetition, before we go anywhere, we are saying is actually done to make sure that the reliability of the results is what is improved. A test was conducted to identify the gas collected in the test tube. So we are seeing the setup here. Test tubes, the retort stent, the Elogia water plant, gas bubbles, and then the funnel is put there. So I always say that in an experiment, I'm going to come up with one question which I'm going to be explaining about an experimental question. An experimental question class, there is no reason which you should also lose any marks there. You should not lose any marks. You must make sure that all the answers are what? You must make sure that all the answers are correct. You understand? You must make sure that all the answers are correct there, and you don't make any mistakes of missing anything. So our question here is saying 2.2.1. Let's just confirm something quickly. 2.2.1, formulate a hypothesis. So I'm saying I'm going to come with a question which I'm going to be explaining about an experimental question to say which one can we say that is the control of the investigation? Why do we need a control in an investigation? What is the dependent and independent variable? And also how to improve the reliability of the results. All those things, I'm going to bring them on one experimental question I will expand and answer the various questions, I mean, that would arise or that you should know. So having said this, I'm sure that 2.1.5, you can add as many points as you can, but I just wanted to give you the direction of what you should also take note of with that answer. There is quite a lot of other points to add. So what we are saying here is that a hypothesis is an educated guess of an investigation. When you're asked to give an hypothesis, a hypothesis actually draws or it provides a guideline towards an investigation, but by giving an answer before the investigation is done. So what we are saying is that when an investigation is being done, we give a hypothesis as to say that is the probable reasons or the probable answer to the investigation that will be carried out. So in this case, what we can say is that the investigation is done, and then we need to look at this to say what can be the hypothesis based on this. So was conducted to determine the effect of light intensity on the rate of photosynthesis. So in this case, what you need to do is that your hypothesis is an answer that is in line with with the investigation that it was conducted to determine the effect of light intensity on rate of photosynthesis. So what I can say is that on that I can maybe say that rate of photosynthesis decreases with increasing light intensity. That is an hypothesis that I can give to say rate of photosynthesis uh, actually decreases with the intensity of light, or rate of photosynthesis decreases with increasing light intensity as one of the hypotheses that I can give. I can say one, rate of photosynthesis decreases, it decreases with increasing light intensity, decreases with increasing light intensity, with increasing light intensity. That is one of the, I can say rate of photosynthesis decreases with increasing light intensity, or I can say it increases, rate of photosynthesis increases with increasing light intensity. I can say it decreases or it increases with increasing light intensity. Or I can as well say that the light intensity has no effect on photosynthesis, as also another point that I can say. Light intensity has no effect on photosynthesis. So I'm saying when you are asked to give a hypothesis class, when you are asked to give a hypothesis, you actually give an answer which is in line with what is to be investigated. So which I said here that experiment was conducted to determine. I'm saying my hypothesis is on this part to determine. An investigation was conducted to determine. It was conducted to determine the effect of light intensity. This is where my hypothesis is actually coming from here. My hypothesis is coming from the reasons of the investigation. So I would give an answer to the situation or to the experiment that is to be conducted. That is what I'm saying. I'm saying hypothesis comes from what is to be investigated. From what is to be investigated. Therefore, it is actually an answer to the investigation. Hypothesis is an answer to the investigation. It is an answer to the investigation. That is one thing that you must understand. It is an answer to that what is being investigated. So let's quickly just go to the next question. Identify the independent variable in the experiment. Identify the independent variable in the investigation. So the independent variable, again, you find it there in the what is being experimented to determine the effect of light on rate of photosynthesis. So there is effect of light, and also we are saying light intensity versus rate of photosynthesis. So the question is saying, what is it that it wants? It wants you to say, identify the independent variable. The independent variable is light intensity. Light intensity is the independent variable that is in question here. Light intensity. Light intensity is the independent variable because photosynthesis is depending on the light intensity. Light is affecting photosynthesis. So the independent, it is what the if I'm saying the independence is light intensity to determine the effect of light on rate of photosynthesis. So photosynthesis is affected by what? By light intensity. So photosynthesis is the dependent variable if they would ask us that. 2.2.3, how was the independent variable measured in this experiment? How was the dependent variable measured? Question 2.1.3, how was it measured? The dependent variable in the experiment. It was actually measured through what? It was measured by what? By counting the number of bubbles released per minute. Counting the number of bubbles. Counting the number of bubbles. The number of bubbles produced per minute. By counting the number of bubbles produced per minute, and then name the gas release during name the gas release as bubbles. Here is it. We are saying obviously you see that the gas that is released as bubbles, we can give it like this to say this is the gas. Obviously, where is the gas coming from? The gas is coming from the water plant. So what is the gas that is produced from the plant? This is what it is, oxygen. It is oxygen gas here. It is oxygen. Oxygen is the gas that is produced here. Oxygen. State two variables that should have been kept constant during this experiment. Two variables that could have been kept constant during this experiment. We can say one, uh, we can talk about temperature must be kept constant. We can say temperature. We can talk about the voltage of the bulb. Voltage of bulb, which we are talking about. That is what the current 3 We can also talk about plant species must be the same. Plant species. Plant species must be the same. We also need to make sure that what is it? The amount of carbon dioxide. Amount of carbon dioxide. Amount of carbon dioxide. We can also talk about the person counting the bubbles. I'm sure that you know when we are doing investigations, ne, we mustn't change the people who are responsible. So person counting the bubbles should be the same. Person counting the bubbles. Person counting the bubbles must not change. I can as well maybe talk about the I may also maybe want to talk about the purity of the water used. Purity of water used. So these are the variables that can be kept constant during the investigation. As you can see, that temperature must be the same. Plant species, we mustn't change them because we need to know how this plant is releasing the carbon, I mean the oxygen, the amount of carbon dioxide also supplied there, the purity of the water also, the person counting the bubbles, and eventually I can also talk about the bulb light, which is the current. You understand? I hope that we are making sense and we are still together. Question 2.6. 2.6. Question 2.6 is saying, why was a pinch of bicarbonate soda added to the beaker at the start of the experiment? We are told that bicarbonate soda was added at the start of the experiment, actually to decrease the concentration of carbon dioxide in the water. I'm sure we can agree together that it was added uh, to increase the concentration of carbon dioxide, to increase the concentration of carbon dioxide, concentration of carbon dioxide in the water, to increase the concentration of carbon dioxide in the water. That is why we add the what? The carbon, I mean the pinch of bicarbonate soda. Describe the test conducted to identify the gas in question 2.2. It is to identify, we are saying to identify the that gas produces oxygen. Oxygen, how do we test it for oxygen? So it is oxygen, it tested by what? Uh, it is tested by the glowing splint. The glowing splint bursts into flame or it grows brighter, which indicate that there is oxygen given off by the plant. So we are saying oxygen is tested by a glowing splint. A glowing splint bursts into flame when it is exposed where there is when it's exposed next to oxygen, it bursts into flame. or other, we can say that it relights. It relights, that is, glows brighter to indicate that oxygen is produced by the plant. To indicate that gas produced by the plant is oxygen. To indicate that the gas produced by the plant is oxygen. When we are testing for oxygen, we use a glowing splint. It bursts into flame, it glows brighter, it relights whenever oxygen is present. And then that is question 2.7. That is question 2.7, glowing splint relights. Question 2.80. The table below shows the number of bubbles released by the Elodia plants when the light source was at different distance. It is showing the number of bubbles released by the plant when light source was at different distance between the conduit and light source, and the number of bubbles produced. So our question is asking to say, calculate the percentage increase in the number of bubbles when the light source was shifted from, when it was shifted from 1.0 to 0.5 meters. Show your working. So when the was shifted from 1.0 to 0.5, so what we are saying is that the difference is that we have got from 1.0 it was 80, and then to 0.5 the number of bubbles is 28. So we want to check the percentage increase in the number of bubbles. So what we do is that it is 28 minus 80 as to say the difference, and then this will give you 20, and then percentage increase is going to be the difference over the initial, which is going to be 20 over 80. 20 over 80 times 100 over 1, which is going to give you something like 250%. Our final 0.5, the bubbles produced was 2080. Before it's 1.0, it was 80. So 28 minus 80, we get 20. We get 20, which is given there. And then 20 over the what? The initial multiplied by what? That is the increase. 20 over initial multiplied by 100, because they want the percentage. Explain how the result obtained in the experiment is beneficial for greenhouse farming of crops. How the result obtained is beneficial for greenhouse farming of crops. How is it beneficial to greenhouse farming of crops? One, it actually enables farmers to adjust the light to its optimal level when farmers are doing greenhouse. One, It enables pharmacy to adjust the light to its optimal level. That is one, which we are saying that is done in order to increase the rate of photosynthesis. So as to increase the rate of photosynthesis. So light can be adjusted to its optimal level so as to increase the rate of photosynthesis. So as to increase the rate of photosynthesis. Also, another thing is that by increasing the rate of photosynthesis, the farmer is able to what? To increase his yields to their maximum. By increasing rate of photosynthesis, the farmer can as well increase his yields. The farmer can as well increase yields to their maximum. You see, it enables farmers to make sure that maximum yields are produced by ensuring effective photosynthesis. When effective photosynthesis takes place, obviously we are to produce the best because the plants are producing or manufacturing their food to the fullest. You understand? So we can actually go to the next question. Represent the main one of the main structures found in the human digestive canal. One of the main structures that are found in human digestive canal, and then we are given that diagram there, 2.3.1. Identify the structure shown in the diagram. Identify the structure shown in the diagram. What is this structure showing? What is this structure shown here? It is actually a villus. The question, it shows a villus here. And then the next question is saying, in what organ in the structure mentioned, in what organ uh, in what organ is the structure mentioned above found? The villus is actually found in the what? In the small intestines, which we can say the ileum, the duodenum, or what. So we can just say in the small intestines. Small intestines. It is found in the small intestines. Small intestines. Others might want to be very particular, which you can just say that it is the ileum. One can say also maybe It is the duodenum, or someone can also say that it is the what? The jejunum. But still, we are talking about the small intestines. The villus is found in the what? Is found in the small intestines. What is the function? The function basically here is to increase the surface area for what? For absorption of digested nutrients. It increases the surface area. Surface area for absorption. It actually increase the surface area for absorption of digested nutrients. Absorption of digested nutrients. And then define the process in which the substance may move into the structure. Describe the process. Describe the process. Okay, define the process 2.2. Define the process by which substance may move into this structure. Actually, the process, define the process. We are just supposed to define what? Diffusion. Define the process. The process is diffusion, class. Diffusion. We can talk about diffusion, which is the movement of molecules from region of its higher concentration to their region of lower concentration. Movement of molecules from their region of higher concentration, from their region of higher concentration to lower concentration. That is diffusion. So diffusion is the movement of molecules from their region of higher concentration to region of lower concentration. That is one process through which substances may move into the structure. We can also maybe talk about also active transport or active uptake, which is the movement of molecules with the assistance of what? The movement of molecules with the assistance of Calea molecules against the concentration gradient from higher concentration to lower concentration using energy. So in terms of this, don't forget again, I'm saying you can talk about diffusion, or you can as well talk about what? Active uptake or active transport, where molecules move with the assistance of what? Move with the assistance of carrier molecules against the concentration gradient uh, from region of higher concentration to region of lower concentration using what? Using energy. That can also be one other answer that you can give besides diffusion. And then the next question, 2.3.5, 2.3.5, explain how the structure shown in the diagram is structurally suited to perform its function. How the structure is structurally suited to perform its function. I can give so much few points there. One, it is actually thin. Actually we are saying the thin columnar epithelium facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. Its diffusion of nutrients facilitates its diffusion of nutrients. it facilitates its diffusion of nutrients. Easy diffusion, like we said, the movement of nutrients from higher concentration to lower concentration is diffusion. Is diffusion of nutrients. And then two, we can also need to understand that it provides, I was saying, provides large surface area so that more nutrients are absorbed in a faster pace. So that more nutrients are absorbed in a faster pace. And then three, what we can also add is that it consists of many blood capillaries. And they like to for faster transport of nutrients away from the site of what? Of absorption. That's the last point. It consists of many blood capillaries for faster transport of nutrients away from the what? From the site of absorption. Faster transport of nutrients away from the site of absorption. Those are few points that we can give as to say how is our question to be answered? The suitability for absorption. How is the villus structurally suited to perform or to facilitate with absorption or active uptake? Those are the few answers that we can, few points that we can give there. Let's just quickly go to the next question. The diagram below represents an experiment set up by a group of learners. All three tubes were exposed to light for 3 hours. The hydrogen carbonate was used as an indicator to test the different levels of carbon dioxide test in the test tubes. Hydrogen carbonate indicator turns purple in the presence of small amount of carbon dioxide, yellow in the presence of large amount of carbon dioxide, orange in the presence of moderate amount of carbon dioxide. So we have got these stoppers there: green leaves, hydrogen carbonate, hydrogen carbonate indicator, and then the bag of live worms, which are to produce what? The carbon dioxide. And then we are given that the result of the experiment is in the table below. The result of the experiment are given as in the table below. And then color of the indicator at the start of the experiment, we have got test tube A, orange, 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 and then all of them meaning to say that they were orange. Color of the indicator after 3 hours, it was purple, yellow, and also orange. as according to the terms that are given there. We are saying purple in the presence of small amount of carbon dioxide, yellow on large amount, and then orange on moderate amounts, as is given in what? In these various specifications or conditions that are given there. So the question is saying state a reason for closing each test tube with a stopper. Why is the test tubes closed with a stopper? It is actually the stopper is there to prevent air from entering the test tube, as it would alter the what? As it would alter the experiment results, or also to stop the gas from entering or leaving. That's what we can say. As you can see here, the stopper here, the advantage or the reason for the stopper, these stoppers here is actually is to prevent or stop the air from entering or leaving the test tubes. So we are saying we need not to allow air to enter or leave the test tubes so that the results will be correct. So that results of investigation will be correct. That's one thing that you should understand it there. The stoppers are actually given there. The stoppers are actually put in order to stop air from entering or leaving the test tube so that the result of the investigation will be correct. And then question next is saying, explain why the indicator in test tube B becomes yellow after 3 hours. The indicator in test tube B, it actually became yellow after 3 hours. As you can see there, test tube B, bag of live worms. Test tube B has got bag of live worms that are there. So it means that they are what? They are producing carbon dioxide there. So what you need to understand is that it contains, because B contains live worms, B contain live worms which are producing large amount of carbon dioxide over time. Live worms producing large amount of carbon dioxide, which are producing large amount of carbon dioxide, over time. I'm saying they contain live worms producing large amount of carbon dioxide during cellular respiration, which causes the indicator in test tube to what? To become yellow. Carbon dioxide during cellular respiration causing the indicator in B to become yellow. The indicator in B uh, in B to be yellow. That is what is happening there. And then what is the purpose of test tube C in the investigation? Test tube C, it does not change. Test tube C does not change. Test tube C does not change. What is the purpose of test tube C in the investigation? I always say that when an experiment is set up in unmanipulated setup, it means it is the control of the investigation. It is there for comparison purposes. So it is C was set up as a control of the investigation. 2.4.3. Let me just write it below here. 2.4. C was set up as a control of the investigation. It was set up as a control of the investigation. What we mean to say is that it helps in determining whether the factor under investigation was actually the one that caused the change. As a control of investigation, that is to indicate to indicate to indicate to indicate whether the factor under investigation actually is the one that causes the change. The factor under investigation was actually the one that caused the change. Under investigation is actually the one that caused it is actually used for comparisons reasons. We compare to see if the results are correct and what might be the causes of those changes. And then question 2.4.4, question 2.4.4, question 2.4.4, explain why the color of the indicator would change to orange or yellow if test tube A is placed in a dark cupboard for the entire day. If test tube A is placed in the dark cupboard for entire day, you see that test tube A, it has got green leaves. And then we are saying test tube A has got green leaves. So what the question is saying is that explain why the color of the indicator would change to green or orange if the test tube is placed in a dark cupboard for the entire day. So what we need to know is that one, no photosynthesis will take place due to lack of what? Due to lack of light. That is one first thing that we need to understand. No photosynthesis will take place because it is in a dark place, will take place due to lack of light. No photosynthesis will take place due to lack of light. Two, you need to understand that the green leaves continue to respire at a rate slower than what? Than animals. We see there are green leaves there. The green leaves they continue to respire at a rate that is slower than animals. That is one thing that you should understand. As a result of that, we also understand that hence release small amounts of carbon dioxide, and later the amount of carbon dioxide increases due to what? To gradual accumulation. They will release they will release small amount of carbon dioxide, and later carbon dioxide increases due to accumulation. That is what you should understand with regards to this question 2.4.4. Explain why the color of the indicator would change to orange or yellow. If test tube A is placed in dark cupboard for the entire day, this number 2.4.4, as you can see, these are the possible reasons which are causing that. These are the possible reasons that you should take note of. And then let's just quickly go to the next question. The diagram represents the process of breathing in humans. It indicates the process of breathing in humans. The diagram represents the process of breathing in humans. As you can see, the chest cavities that are shown in this question there. So we need to understand something here to say that there is also, I think, a question that we also answered that is similar or more or less synonymous to this one with regards to the changes that takes place in inhalation and exhalation. So here we are given that this is the process of breathing that is shown in two diagrams, diagram 1 and 2. So the name the breathing muscle represented by D. Name the breathing muscle that is represented by D. D is representing the what? The diaphragm. D actually represents the diaphragm. D represents the diaphragm. D represents the diaphragm. And then also found muscles that are found at C. Muscles found at C. What muscles are they? Muscles at C. These muscles are called intercostal muscles, and we can sometimes also call them the external and internal intercostal muscles. It's called intercostal muscles. which sometimes they can say internal and external intercostal muscles. We are saying they are called intercostal muscles, which we can say internal or external intercostal muscles. But anyway, the answer remains the same. And then which diagram 1 or 2 shows exhalation? And then you must give a reason for that. Which diagram shows exhalation? Which diagram shows exhalation? Which diagram represents or shows exhalation? Diagram exhalation, it is represented by diagram 2. Diagram 2. It is diagram 2 class. It's diagram 2. And then what are the reasons for that? What are the reasons for why we say it is diagram 2? Diagram 2 shows exhalation. Reason 1, the diaphragm is relaxed. The diaphragm actually is relaxed and therefore restores its original dome shape. The diaphragm is relaxed. The diaphragm is relaxed in B. And therefore restores its dome shape. It restores its dome shape. What else can we say? It restores its dome shape. And then also we can see also in diagram B, exhalation can be correct in diagram B because the size of the chest cavity is substantially reduced, as well as the size of the lungs. Also, the size of the lungs is also what it becomes smaller. The size of the lungs becomes smaller. You know, when we breathe out, the lungs they become smaller. And then I said again, lastly, is that the chest cavity is substantially reduced. Is substantially reduced. The chest cavity is substantially reduced. So that is why we are saying it is showing what? It is showing diagram 2 is showing exhalation. Exhalation is taking place on diagram what? On diagram 2 because of those fundamental observable points that I'm giving there. So next question 3.1.4, identify the letters representing the air pathway where coronavirus would settle and multiply before gaining entry into the lungs. 3.1.4, identify letters representing the air pathway. Letters representing air pathways where coronaviruses would settle and multiply before gaining entry into the lungs. So I'm saying this obviously it is the pathway, it is A to B. They can move from A to B. Remember, we heard that quite a lot of times during COVID-19 that in the nostrils and also on the trachea, which you'll find out quite often that people were told to drink warm water and so forth. So what I'm saying is that the pathway is A to B. It is A to B, and then 3.1.5, 3.1.5. If the coronavirus infects the thin inner lining of the alveoli, large amount of mucus and fluid are secreted as a reaction. This substantially lowers the efficiency of gaseous exchange. This condition is known as pneumonia. Explain why there is substantial increase in breathing rate and heart rate amongst the patients who suffers from pneumonia. Uh, Question A, why is that substantial increase in breathing rate and heart rate from people suffering from pneumonia? So one, the point is that the accumulation of fluids and mucus in the alveoli drastically reduces the rate of gaseous exchange. One, accumulation of fluids and mucus in the alveoli actually drastically reduces or strongly reduces the rate of gaseous exchange. It reduces the rate of gaseous exchange, and then actually this causes an increase in the carbon dioxide level and drop in what? In the oxygen level in the blood, resulting in drop of carbon dioxide, drop in, we are saying resulting in an increase of carbon dioxide, in increase of carbon dioxide, increase in carbon dioxide levels, and a drop in oxygen, and a drop in oxygen level in the blood. And then what happens is that the high level of carbon dioxide stimulates medulla oblongata to send impulses to breathing muscles and heart muscles. That's number two. High levels of carbon dioxide stimulates the medulla oblongata to send impulses to send impulses to breathing muscles. Breathing muscles and also heart muscles. That obviously would cause an increase in the rate and depth of breathing and rate of heart rate, which causes an increase. It causes an increase in heart rate and depth of breathing. And depth of breathing. It causes an impact. It causes an increase in heart rate and depth of breathing, as you can see here. And then what is happening that, or what is causing that, is that it's an attempt to restore carbon dioxide oxygen balance in the blood, in an attempt to restore carbon dioxide and oxygen balance in the blood. That is one thing that you should take note of. And then let me just go to the last question B. Question B. Explain why patients require a ventilator to maintain life. Explain why patients require a ventilator to maintain life, which is a machine that actively pumps highly concentrated oxygen directly into the lungs of the patient through an attached tube inserted to part B, to part labeled B, through the part labeled B, which is this one. So what we are saying is that why would patients require a ventilator to maintain life? Why the need of ventilator? Question B. Why a ventilator? Why a ventilator to maintain life? Why a ventilator to maintain life? That's the question. So we are saying a ventilator, actually, it is a machine that actively pumps highly concentrated oxygen directly into the lungs of the patient through a tube attached to the trachea. That is part labeled B there. So what we are saying is that we need to understand that one, the points are: one, we are saying reduced rate of gaseous exchange due to pneumonia leads to steady drop in oxygen in the blood. That is first point. One, reduced rate of gaseous exchange. Rate of gaseous exchange. Rate of gaseous exchange. Reduced rate of gaseous exchange. which is due to pneumonia, actually leads to a drop in what? In oxygen in the blood. It leads to a drop in oxygen in the blood. And then what happens therefore is that active pumping of oxygen is required to raise the level of oxygen. Therefore, active pumping is required to restore the level of oxygen, in blood. You understand? So to maintain the optimal rate of cellular metabolism, also it is supposed to be on life support. Number 3 or number 2, to maintain optimal rate of cellular metabolism, to maintain optimum or optimal rate of cellular metabolism. To maintain an optimum rate of cellular metabolism, and also to generate energy for the proper functioning of vital organs so as to maintain life. Ventilator, we are saying it's also used to generate energy. Generate energy for the proper functioning of vital organs. Proper functioning of vital organs so as to maintain life. So a ventilator is actually used for reasons such as this one that I'm mentioning here. The reasons that I'm mentioning here are quite relevant with regard to the use of a ventilator. It is actually to maintain proper functioning of vital organs. So therefore, life is not lost. You understand? Let's just quickly go to the next question. A diagram that represents lactic acid levels in the blood for a long distance runner. Graph of lactic concentration in the blood. We are given that the concentration in arbitrary units is given there, and then time in minutes is given there, start and end of the race. Start and end of the race. The first question is saying, name the type of respiration that takes place in the cells of long distance runner 15 minutes after the race has started. The type of respiration 15 minutes after the race has started. It is actually, we are saying the type of respiration which is taking place there is anaerobic respiration. anaerobic respiration. It is anaerobic respiration. Others, we can as well maybe mention it as what? As lactic acid fermentation. Lactic acid fermentation. And then question B is saying name the site where lactic acid accumulates during this process. The site where lactic acid accumulates during this process, it actually occurs in the what? In the skeletal muscles. The skeletal muscles. And then for how long did for how long did the lactic acid concentration continue to increase after the end of the race? For how long did the lactic acid concentration continue to increase after the end of the race? After the end of the race, end of the race is ending there. So for how long did it continue? For how long did it continue to? For how long did the lactic acid concentration continue to increase after the end of the race? So end of the race, it the race ended at 20. Maybe I can say it is at 20 where the race is ending. It is at 20 somewhere there. So for how long did it continue to increase? So we can say that it continuously increased uh, for minutes is from 20 up to somewhere here. That is where it increased. So how many minutes can this be? I can say 510. We can say it's from 20 to 25, it's 510, maybe something like 12 minutes after the end, because the end was 20 and then it continuously increased from up to 30, I guess, or maybe say uh 510, 30, maybe up somewhere here. I don't, let me just try to see. So which is maybe 15, maybe 50, not maybe 13 minutes. It can be 13 minutes. 13 minutes. What you just need to do is that we are talking about from the increase after the end of the race. So the race ended at the race ended actually it ended somewhere there. So that is where the race ended. So the race ended somewhere there, which we are saying it is. So that is the calculation that you just need to take note of, to say you are looking at this time. You are looking at that time to say how many minutes can that be. This is the time that you are calculating. That's the time that you are calculating, class, from here up to there. How many minutes are these? You can up to say maybe 14, 14, 15, they might be correct there. 18, 14, 15. Explain why there is an increase in the lactic acid concentration in the blood. why is there an increase in the lactic acid concentration in the blood? Explain why there is an increase in lactic acid concentration in the blood. It is caused because of what? One, it is caused by intense physical activity leads to an increased rate of cellular respiration. That is one point. We are saying intense physical activity, increase in physical activity, the increase in physical activity, which also leads to increased cellular respiration, which causes an accumulation of carbon dioxide and also decrease in the level of available oxygen due to insufficient what? Ventilation. Therefore, only glycolysis takes place in that regard. And in the absence of sufficient oxygen, the pyruvic acid molecules are actually converted to lactic acid, which is stored in the what? In muscle tissues. So that is the reason why you see that there is what? There is an increase in lactic acid, an intense increase in physical activity, actually leading to increased rate of cellular respiration. That actually causes an accumulation of carbon dioxide and also decrease in the level of available oxygen due to insufficient what? Due to insufficient ventilation. In that regard, we therefore have only glycolysis taking place in the absence of what? Sufficient oxygen, and hence the pyruvic acid molecules are what? Are converted to lactic acid, which is stored in the muscle tissues. That is the reason why there is an increase in what? In lactic acid. And then the last question, question 3.25, explain why a long distance runner who is suffering from severe muscle cramp is advised to rest immediately. Why is he advised to rest immediately? One, no physical activity occurs during resting. No physical activity occurs during resting. No physical activity occurs during resting. Therefore, less glucose is restored to what? Less glucose is required to be oxidized. Hence, less glucose is required to be oxidized. Which therefore, in that regard, we are saying, therefore, the demand for oxygen decreases, or is decreased. The demand for oxygen is decreased, and then carbon dioxide is expelled from the body gradually. Oxygen balance will be restored. Another thing is that the lactic acid stored in the muscles will be converted back to what? Will be converted back to pyruvic acid, enabling the cells to complete the process or the Krebs cycle, and hence oxidative phosphorylation will take place. So that is why the person is required to rest. That is why the person is required to rest to restore the balance of carbon dioxide and oxygen, which is in that regard enabling the lactic acid stored in the muscles to be converted back to what? Back to pyruvic acid. That enables the complete process of what? Of Krebs cycle and what? And oxidative phosphorylation. Question 3 point. Next question. Read the extract below. What is the extract? Is hypermetria. Hypermetria is the medical term used to describe having too much sodium in the blood. Sodium is an important nutrient for proper functioning of the body. Most of the body sodium is found in the blood. Hypermetria can occur when there is too much water loss or too much sodium gain in the body. The result is too little body water for the amount of total sodium. What is this condition? What is that condition? What is that condition? It is actually a condition characterized by excess sodium in the blood. A condition characterized, which is characterized by excess sodium in the blood. Characterized by excess sodium in blood. That is the condition. With reference to the extract, state two possible reasons for this. According to this, it is caused by too much water loss. Too much water loss. It can be due to too much water loss, or it can also be due to high intake of salt. High intake of salt, which we are saying sodium also. I'm seeing that you can see that too much water loss, too much sodium gain in the body are the factors that are causing this condition. Too much water loss and too much sodium in the body can also be the condition that we are referring to. And then next question, explain why sodium ions are actively pumped out at certain regions of the loop of the hen into the medullary region of the kidney. Why is it like that? Question 3.3. It is because high concentration of sodium creates what? A steeper concentration gradient. Question 3.3. 1. High concentration of sodium creates a steeper gradient. Creates a steeper gradient. And then which actually leads to passive absorption of water. Leading to passive absorption of water. Which is actually causing passive absorption of water from the collecting from the collecting tubules into the surrounding tissue fluid. From collecting tubules into the surrounding tissue fluid. the surrounding tissue fluid in medulla region, basically, that we can say. And then next question, 3.3.4, describe how constant sodium concentration in the blood, in the blood of a healthy human is maintained when the level of sodium is decreased below normal. It's just the balancing of what? The balancing of salt in the body. 3.3.4. Uh, in this case, class, what you need to understand is just you need to explain about the balancing of salt in the body, uh, where we are saying this person is a healthy person when the level of sodium decreased below normal. So how is the salt level is restored? The restoration of salt level, that is what you need to describe here. So the lower level of sodium in the blood stimulates the adrenal gland to secrete and release more aldosterone into the blood. More aldosterone is released into the blood, which actually causes the reabsorption of what? Of more sodium by capillaries at the distal collecting tubules, or less sodium ions are what are excreted from the kidneys. So sodium ions are reabsorbed until its level in the blood returns to what? To normal. That is what is supposed to be understood in. So that is what you need to understand, that the lower level of sodium in the blood stimulates what? It stimulates the adrenal gland to secrete and release more aldosterone in the blood, which actually causes the reabsorption of more sodium by the capillaries at the distal and collecting tubules. Or we can as well say that less sodium ions are excreted from the kidneys. Therefore, sodium ions are reabsorbed until the level in the blood returns to normal. That is what happens with what? Question number 4. And then question 5, explain why the level of antidiuretic hormone remains high in patient who is severely dehydrated with diarrhea and vomiting. So one thing that we need to understand is that diarrhea and vomiting, actually leads to excess loss of what? Excess loss of water and what? And salt. Diarrhea and vomiting, diarrhea and vomiting leads to excessive loss of there is excessive loss of water and what? And salt. Excessive loss of water and salt, and then drop in the level of water in the blood, or causes dehydration, that actually stimulates the hypothalamus to release what? To release more ADH in the blood. It actually stimulates the hypothalamus, to release what? To release more ADH into the blood. That is the reason why there is always remaining, the ADH remains high. That is why it remains high. It's a question showing the changes in blood glucose levels. A healthy person after insulin injection. A healthy person after insulin injection. And then we have got insulin injected there. We have got time and blood glucose concentration, which are shown there. I'm sure you can see nicely this graph there. And then state the amount of glucose that was present in the person's blood at the time of injection. At the time of injection, we are saying the amount of glucose at the time of injection. It is shown here, class 0.7 milligrams per cubic centimeter. At the time of injection, it is at the time injection insulin injected is what 0.7. It's actually 0.7. It is shown there. The amount of glucose at the time of injection. The time of injection is shown on the what? On the graph here is 0.7 milligrams. And then 10 minutes after the insulin injection, 10 minutes. So we have got 10 minutes after the insulin injection. Is it at the half mark of this? That is 10 minutes. Because our time is going like this. So we can say at the half mark of this, we can go up and see. So I can give to say that at 10 minutes, 10 minutes, this is 10 minutes. So the reading can be what? 0.5 milligrams also per cubic centimeter is what is shown there, 0.5. I'm saying you just mark the 10 minutes and check across the graph. Then you can see that is 0.5. And then we have got 0.7. And then the next question is saying, explain the drastic drop in the glucose level of the normal health person after the injection. In this case, just for the sake of time, guys, I'm just going to quickly just explain them. I'm not going to write a lot because of time again. So the drastic drop in the blood glucose level of the health person after the injection, the health person maintains optimum level of insulin and also actually and hence maintains normal blood glucose level. Additional amount of insulin injected converts more glucose in the what? In the blood to glycogen. Because when insulin is injected there, we are saying additional amount of insulin injected converts more glucose in the blood to glycogen, which is stored in the muscle and liver, dropping the blood glucose level drastically. That is why you see, I'm sure I remember that insulin, the function of insulin that is injected, it actually converts glucose. It converts glucose to what? To glycogen. The storage form of glucose. So what is happening is that we are saying that the drastic drop in glucose level is because the person is healthy. Hence, the glucose that is in the body is converted into glycogen. That is why you see that drastic drop there. And then, which is therefore additional amount of insulin injected converts more glucose in the blood to glycogen, which is stored in the muscles and liver, causing the drop in what? In glucose level drastically. And then number 3.25, 4.3, explain how the normal blood glucose level is restored 2 hours after injection. In this case, what you need to understand is that when the level of glucose drops below normal, pancreas, actually pancreas cells, or we are saying the cells of lung organs are stimulated to secrete more glucagon into the blood, which actually causes the muscles and liver to convert stored glycogen into glucose. This raises the level of glucose back to normal. I'm sure I remember this when I gave you the diagram that is uh, increasing and decreasing or balancing of glucose in the blood. Glucose, and then we have got glycogen, and then I said when it goes there, it is what is it is insulin that does that, and then when it comes back here, it is what it is glucagon. So that is what you need to understand, that the conversion processes that takes place in this regard in order to restore glucose back to normal is what, like I said there, to say that whenever the glucose level drops below normal, the pancreas or the cells of lung organs are stimulated to what? To secrete more glucagon into the blood, which is the glucagon that you are seeing here on this diagram. And then, which causes the muscles, which actually causes the muscles and liver to convert stored glycogen into glucose, and then raising back the glucose level to normal. And then last question 3.44. Uh, some diabetes patients collapse due to sudden drop in the blood glucose level. In order to raise the blood glucose level, they are given a concentrated glucose solution rather than starch-containing food to explain the reason for this treatment. In this case, what is happening is that concentrated glucose will be absorbed directly into the what? Into the blood, because glucose is the what? Is the monomer unit of carbohydrates, which actually requires no digestion. That is the reason for this question. Concentration, I mean, the concentrated actually concentrated glucose will be absorbed. It will be absorbed directly into the blood. It will be absorbed directly into the blood. Since, because glucose is the monomer unit of carbohydrates. Which requires no digestion. That is one thing that you need to understand with regard to that question. So, classy, thank you so much, and I hope you be patient with the presentation. It looks long, However, it is because we have got what? We have got we have to do the full question. We have to do the full paper, sorry. So thank you so much for your time and your patience. Keep on following. We are going to get this well and well done. Don't forget next year, grade 12 content is totally loaded and is keeping on coming. More and more is coming. So you are in safe hands. Life science, it is done. Thank you and have a beautiful day. Thank you.